it does, and everybody else can see it, I hope.
We can.
Oh, wonderful. Thank you. Um,
Sure.
so I want to, so that's yet another example of what it's like to have worked with Nebojsa for half of my life. Um, it's a remarkable thing. I also want to thank you for letting me do this. I realized in the first half of today that the amount of chemistry I've learned from Nebojsa again is huge, but actually inadequate. So if I get a chance to come back, I will learn chemistry because I'd like to. So this is the title. I'll take as little time as I can. You all know about SOMers. You sort of know about SomaScan as a proteomic platform. And I arranged my talk to introduce stuff and then to tell you two examples of tissues used to use in a proteomic profiling way to tell you that you can make a gigantic leap into drug targets and treatments in a way that really is about the leap from correlation to causality. That's the talk I'm really giving. Next slide, Nebojsa. Thank you. Oh, this is the one that Nebojsa said, I hate showing this. I don't like it much better than that, but there you go. So this is the same slide Nebojsa showed you, cleaned up on the left where it says protein target. And somomers are slow off rate modified aptamers. You all know that. Well, we've been hanging with this for a long time. And the modifications we do are almost always pyrimidine modifications. We have many more modifications on U than on C. And everybody's talks about click chemistry and all this way of generating additional variations. It was wonderful hearing them. I just read last week Gunter's wonderful longer version of the one you heard today about click chemistry, and it's all terrific. Next slide. This slide will take a nanosecond. This is a slide that says, you know, when you look at co-crystals, they're really something. So the one thing I would say is that you all know, because you've read all of these papers, including Amy's review article that's shown in the bottom, once you start adding things to classic aptamers, RNA or DNA, these kinds of hydrophobic things, you end up with structures that are totally unanticipated. Little motifs that you never would have believed were going to be there. So this is not just additional base pairs, as people are also doing, but really using oligonucleotides to create new shapes and new motifs that you wouldn't have ever believed could happen. The next slide is my favorite slide of my life ever. And it is a slide of one of the co-crystals we have. And it's just, when you look at it, I love to show this to people. Nebojsa and I have probably each showed this slide 7,000 times. And until you get to the mods, the purple things, and then finally the actual, you know, structure of the somomer, the average person cannot tell the difference between the two globular things shown in the beginning, because you don't know which is a protein and which is a DNA molecule. So to me, that is the essence of selects and aptamers, the things that Craig did and Andy Ellington. The essence is we made a business, if you will, out of studying the globular qualities of single-stranded nucleic acids. When we started all this 30 years ago, the world view was that the only globular RNA molecules then were 5sRNA, ribosomal RNA, and tRNAs. That was it. And so they were considered outliers. Nowadays, because of a lot of beautiful work that people have done, there are a lot of globular RNAs. And Selects is really all about globular RNAs, and this picture says that. Next slide. So there is a thing that we've worked on now, we at SomaLogic, and 3 years before that at Nexstar, we've worked on turning somomers now into a proteomic assay. And the details of this were published, A, and B don't matter for this talk. The work is stunning, done by lots of people. I think the first paper that we published about how to do proteomics in an unbiased and broad way had 100 authors. There were so many authors that some of us didn't even know all the authors, even though they were in our building. And so it took a lot of time. In the first 10 years, it didn't work at all because we had some ideas that were dumber than we eventually came to. And today, when you do SomaScan, which is just a multiplex proteomic thing, using a single somomer and kinetics to get the kind of specificity you get out of an ELISA, the present version of SomaScan is 7,000 different human proteins. So we've done about, I'm making this number up a little bit, about 10,000 selections at SomaLogic, which is a lot of selects experiments. And what's really interesting is about all the different ways that people try to make it quicker. I loved Gunter's presentation about a repeat, really, not a repeat, but an extension of what Andy Ellington tried to do for a long, long time ago, which was to automate selects. And we played with that. And there came a moment when we realized that the really slow step of aptamer and somomer development is what you do after you get them. So if it takes us a week or two, and we always do 100 at a time, that week or two is piddling compared to the work that goes on to make sure you got what you wanted. I mean, look at Neboisha's talk today, Neboisha's and Williams' talk today. The work that Amy did was extraordinary, her 25 selections on 5 different targets. But the work that followed is more extraordinary, and it took much, much longer. So in a sense, the speed of selection is not the rate limiting thing to getting to scale. We're at scale now with a 7,000 protein offering, if you will, and that content will grow. You only got to get to 20,000 to do everything. So I expect we will finish that job sometime before long. Next slide. So this is the history. We've done hundreds of thousands of samples, mostly plasma, serum, or urine. Had a lot of collaborations with academic scientists, medical school scientists, biotech companies, big pharma. Some of them are not exactly collaborations, they're customers, but that's okay. They're still friends. There are probably 4,000 or so proteins that are intentionally secreted from cells, have protein sequences that aid their getting out of cells to signal in a paracrine or even in a distal fashion in the body. But you can tell from the number 7,000 and the number 4,000 that we're measuring a lot of proteins that are not secreted. And that's interesting, and that number will grow. We're not yet measuring all the secreted proteins, but that will happen soon. So the question I'd like to pose is, what about tissue samples? Could we do that and learn something a bit more direct, where at least we knew what cell or cells were making and collecting those proteins? So we've done a fair amount of that. Most of it is not published. A lot of it is. And it wouldn't surprise you that if you compared, lung tissue with, breast tumors or whatever you want, any 2 tissues, that the protein concentrations will be different, the relative protein concentrations. And we don't worry about that. I mean, it's just obvious. That's the way it is. That's called differentiation. And our belief is, and I'm going to show you that it's true, that if we did tissue analyses, sometimes we will get close to what's causing either a disease phenotype or something you care about. So that bottom thing is the reason I decided to focus not on the 500,000 samples that we've done at the top of this slide, but on the less than 1,000 samples that we've done from tissue homogenites. Next slide. Naboisha, there you go. So first I want to tell you about the experiments we did years ago. These are mostly old, um, but they're still relevant. They're not wrong, they're just old. About grinding up tumors and looking on SomaScan to see what proteins you find in those homogenates. And one of the very first things we found, which is looks like we did just a thrombospondin 2 assay. That's not true. These data come from a smaller version of SomaScan than the present version. And we were interested in a question that people have asked over and over and over. This is published by a wonderful scientist who's now elsewhere, a guy named Mike Meehan, who did this analysis with a great pathologist who sent us the tissues. And the question was, if you looked in a tumor, what happened to either the adjacent non-tumor tissue and the distant tissue? Did you ever see anything that made you think, wow, this tumor is inducing the cells that are at some distance? So that's what this slide was about. And we found no evidence ourselves. No, we were only looking at a couple thousand proteins at that time. No real evidence of induction. There's a whole literature of induction in healthy tissues adjacent to tumors. So it can't be true that we've eliminated adjacency of any kind. But by and large, you don't see a lot when you do that. Then we get to the confirmation of this. So thrombospondin 2 is an important protein. And in the next slide, again, old data, the very same SOMAmer that was used to see the increased concentration of thrombospondin 2 in tumors was used to stain the tumor. You can do that. You have to keep in mind that when you're trying to stain tissue sections, the SOMAmers are globular and the proteins are globular. And so the experiments that work best are frozen tissues, fresh frozen, rather than formaldehyde fixed tissue, because the fixation often destroys the conformation that the SOMAmers recognize. There's recent work on high resolution microscopy that has been published and that is beautiful, and it was talked about last week. And you can gently fix so that you can still use SOMAmers, but that's a little bit of an art form compared to just using frozen fresh tissue. It doesn't matter for what I'm trying to tell you, which is that thrombospondin was found in the stroma around the tumor, penetrating sometimes into the tumor. And so at first you think, wait a minute, you didn't stain the tumor. Well, that was okay, because in the experiments where we did SomaScan, the stroma was included in what we homogenized. Next slide is the beginning of the money slide, if you will, for this kind of tumor tissue homogenate. So we decided we'd study non-small cell lung cancer. We've been interested in that forever. And so these are the samples we got. We started with 63, which I'm going to focus on in a minute. Then we grew some more. The SomaScan was at the time only 1129 proteins. Then there's data from the 4,000 plex. There's data, as it says in study 3, that from a needle biopsy, you get enough tissue to be able to do SomaScan, which is terrific. And then there are lots and lots more samples that are available. And the question is, what did we find? And this is the beginning of saying sometimes what you find by doing tissue proteomics is interesting, but not maybe therapeutically useful. The second example I'm going to give has a chance of being enormously therapeutically useful. So here's a screenshot of the way we organize the data from SomaScan. Next slide, sorry. Oh, there you go. So this is a complicated slide. This was, and I don't like showing complicated slides because I'm capable of forgetting what I'm talking about, even with simple slides, actually. So this is the very first data set that was done with 63 samples. And it's organized in a funny way. It's organized from left to right, except for the first column, which we'll come back to, by protein that was on the then SomaScan menu. So left to right is just proteins, okay? And if you look up at the top, they're going from column E over by protein. So what is the order going left to right? Remember, we did 63 samples in this first study, 63 samples whose adjacent tissue, healthy tissue, and the tumor were each analyzed on SomaScan. So we were really looking at the ratio of a protein concentration in tumor, essentially divided by the protein concentration in the adjacent healthy tissue, spurred by Mike Meehan's initial experiment with thrombospondin in the thing I showed you. And so the coloring is, if you were down in the tumor for a protein, you could be red. And if you were up in a tumor, you could be green. And there are lots of proteins that are largely ups and lots of proteins that are largely downs. So if you look in the first columns, E and F, which you can read, you can see mostly red for a thing called ager something or another, a protein whose activities I don't know anything about, but it's clearly a biomarker that goes down. And then there's this thrombospondin 2 thing that we already talked about, and it's largely ups. So there's mostly green in that column. The left-hand column, column B, is the column that names the kind of tumor this was. It was a non-small cell lung tumor, but they were different kinds, and they were, so each row is a tumor, one tumor. So when you look up at columns E and F, you see the number 51. What does that mean? It means that 51 out of the 63 samples showed an up in the red for the column E, and a down rather, and an up in the green for column F. So how did we decide what it took to be labeled down or up? And if you look in the upper left-hand corner of the spreadsheet, there's a thing called the threshold. And the number 4 that you see there means that for this screenshot, you had to be either down by 4-fold or up by 4-fold. So to be a green, you had to be, the ratio for the tumor to healthy tissue had to be 4 or more because of the threshold being set at 4. And for the red, it had to be 0.25 or less. The most interesting thing about this, there are many interesting things, is I've spent hundreds of hours staring at this thing. It's ordered by the number 51. The numbers at that is for the samples outside the threshold. And you can see that it was only 51 of 63. That was the best. That means there were 12 out of 63 that were not outside the threshold, not a significant signal to noise to be counted as a biomarker. The more interesting thing, and what is, I think, really the difficulties of, and I'll show this in the next slide in a second, I put a box around 2 adeno tumors that were the same stage, stage 1A, and the same KRAS mutation. So by both staging and genotyping, those 2 people today would be treated similarly. Actually, today they might be treated similarly in a way that would work because Amgen made a drug aimed at this KRAS mutation. And if that were available to everybody, that would be more important than what I'm telling you, which is usually the case. But look at the left-hand side of that red box. It says that for one of those tumors, there were only 3 proteins, 3 out of the many that were measured on SomaScan that were either 4-fold up or 4-fold down. And the same exact tumor type, 1A, and a specific KRAS mutation, same one, same codon, that number on the left is 157. That means that, more than 10% of the proteins were outside the threshold for one tumor, and the other was tiny, almost no tumor differences. And that, I think, is the next slide, Nabush, if you don't mind, if you're still awake. Next slide is kind of a summary of this. What did we learn from this experiment? Tumors can be very different even when they have the same driver mutations. The way I think of tumors is there's an event, it's usually a mutation of some sort in a normal cell, starting a pathway of Darwinian evolution over decades, with high mutation rates being, for me, the most important characteristic of tumors, most tumors, many tumors. And you could think of every host who has his or her own genotype, his or her own environmental, his or her whatever, an independent example of the context within which that Darwinian evolution occurs over 30 years. So maybe it's not so surprising that tumor biomarkers should be very different like I just showed you, for those 2 KRAS identical driver mutations. The biology, the proteomics anyway, is very, very different. And actually, in my mind, the bottom bullet is really what I think is likely to be over the next years the dominant. It's already huge, and I think that drugs like Keytruda that ask your immune system to pay attention to your evolving tumor, your independently evolving tumor, and kill it, is a way of eliminating our deep concern for the heterogeneity of tumors. That's the end of that. I have only one more little section to go. Next slide. So do we have any examples where the target discovery and treatment kind of get your juices flowing? And we do. And this is all unpublished, except for the paper that I'll show you. So this is about human pain. And it's a collaboration with a guy named Scott Fauci, who by coincidence is a scientist in Denver at Craig Hospital. And you have to read this because there's so many shocking things here. Craig Hospital specializes in the rehabilitation of people with spinal cord injuries, paraplegics and quadriplegics, a terrible thing that happens to people. The 15% or so of the people who are paralyzed by an accident got paralyzed in an accident in which they tripped over their dog. And they tripped over their dog and they're injured permanently, even when they fall on a carpeted floor. And this 4th bullet is a shocking thing to learn, which we learned from Scott, who's a friend now. We've been collaborating with him for about 6 years. And because he is the doctor who first stabilizes people who are at risk of dying, it stabilizes their spinal cord injury so it doesn't get worse. And then he would, because he's such a good guy, he would go and visit his patients. And after a month or so, the bulk of the patients who had traumatic spinal cord injuries were complaining less about their paralysis and more about their pain by far. And they would sometimes get suicidal because the pain was so intense. It took about a month to develop, sometimes a little longer, but it's fast. And they needed help from their surgeon. And he was being begged to operate and get rid of the pain. And the pain was often, most frequently, below the level of the injury. So kind of akin to the way we've learned to think about phantom pain in people who have suffered amputations. But this is slightly different. The injury was a spinal cord injury, and most of the people who experience pain are experiencing intense pain below the level of the injury. And Scott, who studied this and started doing operations and figuring out what I'm going to show you in the next slide or two, found the tissue that was the source of the unrelenting, as it says, and severe pain. And the next slide is a summary slide of, there it is right there, a key paper published by Scott. We had nothing to do with any of this. All of this was his work and the small group of people working with him at Craig Hospital. And this paper is worth reading. It's not, I mean, I know you guys now better than I did yesterday. I mean, you know, you're chemists and a lot of chemists. And, you know, this is not the sort of thing, even for me, a biologist, I wish I was more of a chemist, but for me, this is a hard paper to read, but it is filled with stuff. And he was asked to help all these patients, and he found 3 unique patients, as it says in the second bullet, who had intractable pain, spinal cord injuries, SCI, and they had had complete spinal cord transections from their accident. But I'll get there. Nebojsa, are you trying to tell me it's time for dinner? I love it. I'm really almost done. Sorry. These patients were in trouble, and over years, he worked on this problem for 30 years. I mean, he's getting to be not a spring chicken himself. And he found only the complete transections for spinal cord injuries are rare. And he found in his patient group 3 patients who were clearly, completely, cleanly severed spinal cord. So you didn't have to wonder, from what I'm about to tell you, whether the patients were able to get nerve signals to the brain through the damage in the spinal cord injury. That was not possible for these 3 more severely injured patients than the bulk. Scott has treated almost 300 patients with what I'm about to tell you. And he found electrically active tissue in a very narrow place along the spinal column below the break. And when he cauterized that tissue, for these 3 patients in this paper, the patients were relieved of all pain, and it lasted as long as it has been since the operation. So amazing. He cauterized tissue below the break. And we all learned in high school, if you do that, that you cannot signal to the brain because you got to go up the spinal column. And this guy discovered that you don't have to go up the spinal column. And now he has fMRI data and all that kind of stuff that shows that when he does this surgery, or this ablation, he changes the fMRI signal in the brain with a completely separate spinal column. Next slide, please. So the next 2 slides, you don't have to really stare at it. We'll just jump right through them. One is a slide of electrically normal tissues. And, you know, he sits there with a little amp meter, and he just goes up and down the spinal column until he finds something that looks like the next slide. And the next slide says, here I am, next slide, right there. And you can tell with his little thing that he's poking around in your spinal column, but he found after 20 years of futzing around that tissue is different and has no way for those signals to get to the brain because the spinal column is severed. And so what he did with this information, next slide, is he would find those tissues And he has an idea about how it works, that those electrically hot tissues are sprouting nerve things that are going up beyond the severed spinal column and diving back into the, the spinal column above the break, essentially making a short circuit around the break, the spinal cord injury, so that you can reach the brain. This idea, data are consistent with that. How it does that, of course, is unknown. Okay, next slide. There you go. So he knows it's causal. Why does he know it's causal? Because when he ablates it or removes it, the pain goes away. The end of story. That's a definition of causal. And he's done now 250 patients, and about 85% of them have been responsive by no pain. Okay, last slide, next slide. So we did the obvious. He came to us a few years ago and he wanted us to do SomaScan on the tissue. We did it. He got electrically hot and electrically cold, and we compared them. And the next slide is the kind of half of the money slide. When you look, this was when SomaScan was smaller. So we've been doing this with him a long time. And you see this little upper quadrant section in a volcano plot. These are the ratios. And you see a bunch of proteins that are overproduced significantly. And that number today, because we've run all these samples on the 5,000 plex, so the number of proteins on this figure in that red box would be about 60. So you should remember that, 60. That's about right, plus or minus 1. And he noticed that one of them was a protein, this protein called SV2A, that was correlated with being correlated with pain. And he realized there was a drug aimed at it that you could buy for a different purpose. Next slide. And when he did that, he's a little lab and little group in Denver. So he's only done 5 people, but 5 is better than none, using the drug that is already approved for another indication that is an inhibitor of SV2A. This is trying to get correlation to causality, a very difficult jump. And this thing says, my God, it worked. The first thing he tried worked. That's all it says. Wow. Kind of 4 for 5 or 5 for 5, depending on whether you think one patient did or did not really respond. But the worst thing is the next to the bottom bullet. This is the America. This is the United States. And his insurance company with a person who had suicidal pain was refused payment to take this drug. Those of you who are in Europe should look at that and think, God, they are helpless and hopeless. Next slide. We're all done, Naboisha. The next slide. So this is what we learned. Spinal injuries can cause unrelenting pain soon after injury. It takes some time for cell growth, I think. The cause here is not the way you think about tumors, starting with mutations and taking 30 years to develop. This is instantaneous in biological terms, takes a month. We've obviously spent years now looking at these 60 proteins, years, trying to figure out what should happen next. It's clear that this SV2A inhibitor is a part of it, but I think it would be crazy from what we already know to not think of this as going to require multiple drugs aimed at multiple pathways to get treatment that is not Scott's surgery, because Scott can only do these surgeries take a day and he can't do enough. And you have to worry or wonder whether repurposed drugs are the new ones that will come from, say, the 45 or so that don't have drugs of the 60, whether there will be enough clinical trial activity and development of new drugs to make this into something of value for people, even beyond spinal cord injury patients. And I'm done. Thank you for listening. Thanks a lot. And thanks, Naboisha, for inviting me.